Welcome to the deep dive. Even with all the incredible medical progress we've seen, there's still this huge challenge hanging over global health.
hmm
Developing an effective HIV vaccine, it's still just a massive priority, isn't it?
Absolutely. I mean, the treatments and prevention methods are amazing now, truly remarkable. But
Yeah.
yeah, vaccine is really the key, the ultimate goal to properly end the pandemic.
And one group right in the thick of it since, gosh, 1996 has been IAVI, the International AIDS Vaccine Initiative.
That's right. A major player for decades now.
And just to get a sense of the scale here, in 2023, we were looking at almost 40 million people globally living with HIV/AIDS.
40 million. And still seeing about 1.3 million new infections every year,
Hmm.
which is,
It
staggering.
really is.
And what's really sobering is that almost a quarter of those people, about 23%, they don't have access to the treatments that can suppress the virus.
Which means they can still transmit it.
Exactly. And then there's the cost, the financial resources needed for the global response.
Yeah.
It's huge, projected to be around $29 billion by 2025.
Wow. Okay, so for our deep dive today, our mission is pretty clear, right? We want to unpack why making an HIV vaccine has been so incredibly hard, and then explore the really promising new strategies that IAVI and their partners are working on.
Exactly. We'll dig into the science, the hurdles, what made it such a tough puzzle, and also the innovations that are, genuinely offering hope now.
Right. This is for anyone wanting to get a real handle on, well, one of the biggest global health challenges out there.
Precisely.
Okay, let's dive in then. The hurdles. Why has this been so difficult? We've cured or vaccinated against so many other things. What makes HIV different?
Yeah, that's the core question, isn't it? And it really comes down to how uniquely tricky HIV is. For starters, the virus is just incredibly good at evading our immune system.
How does it do that?
Well, it essentially wears this molecular cloak.
Yeah.
It's covered in these dense clumps of sugar molecules,
Mm.
glycans technically,
Mm.
and they basically hide the parts of the virus that antibodies would normally latch onto.
So it's like it's got an invisibility shield almost?
That's a great way to put it, yes. And as if that wasn't enough, HIV is, well, it's incredibly diverse genetically. It mutates constantly.
Constantly.
Oh, yeah. Not just between people, but even inside one person over time. So you can't just design a vaccine for one HIV. It needs to work against this huge shifting cloud of different strains.
That sounds almost impossible, like trying to hit a swarm of gnats with one fly swatter.
It's a very good analogy.
Yeah.
It really is that difficult. And then there's another really fundamental problem.
Okay.
Unlike lots of other diseases where maybe you get sick, your body fights it off, and then you're immune. Nobody has ever naturally cleared an HIV infection. Ever.
Nobody. Wow.
No. So scientists don't have that natural model of protective immunity to study, to learn from. They're basically trying to design immunity from scratch without a clear blueprint of what success even looks like naturally.
Okay, that's a huge hurdle. Building something without knowing exactly what the finished immune response should be aiming for.
Exactly. And sort of the final piece of this puzzle, HIV directly attacks the immune system itself. It goes after the key command cells, the CD4 T cells.
The very cells meant to fight it off.
Precisely. And it doesn't just kill them, it integrates its genetic material right into their DNA. So any vaccine needs to provoke an incredibly powerful, incredibly fast immune response to stop the virus before it gets into those cells and hides.
So your standard vaccine approaches just weren't cutting it against all that.
No, they really struggled against that combination of evasion, diversity, lack of a natural model, and attacking the immune system itself. It required a whole new way of thinking.
Right, so that leads us to this new era you mentioned, the discovery of these broadly neutralizing antibodies.
bnAbs.
Right. What are they and why were they such a big deal?
Yes, bnAbs. This discovery, starting around 2009, was a genuine game changer. Researchers found these very special, very potent antibodies in a small number of people who'd been living with HIV for years.
Found them where?
All over, actually. Africa, India, Thailand, Australia, UK, US. And what was amazing was that these weren't like normal antibodies that might only target one specific HIV strain. These could neutralize a broad range of different HIV variants.
So like a master key instead of just one specific key.
Exactly. Think of them as super antibodies. They gave scientists the first real glimpse of what a truly effective immune response against the diversity of HIV might actually look like.
Okay, that sounds huge. So who found them?
Well, IAVI's Neutralizing Antibody Center, the NAC, that's a collaboration with Scripps Research and other key partners worldwide, They were absolutely crucial in isolating these bnAbs and figuring out how they work.
And they worked in the lab.
Yes, lab studies, in vitro studies, showed clearly that some of these bnAbs could block HIV from infecting cells. That was a major turning point. It offered a new target for vaccine design. Could we get the body to make these?
Okay, so lab work is one thing. Did they test this idea in people? You mentioned a 2021 study?
Yes, exactly. That study was really important. It showed that giving people infusions of these bnAbs could actually protect them from acquiring HIV. Proof of concept.
Wow. So it worked.
It did work, but with some significant caveats. You needed a good match between the bnAbs being infused and the specific HIV strains the person was exposed to.
Ah, okay.
And you also needed pretty high levels of these antibodies circulating in the blood. So while it proved bnAbs could work in humans,
It wasn't really a practical vaccine strategy in itself, just giving infusions.
precisely, you can't just keep infusing people constantly.
Yeah.
But it was critical evidence. It showed bnAbs can block infection, strongly supporting IAVI's strategy of designing vaccines to make our own bodies produce them. Gotcha. Okay, so the goal became design a vaccine to elicit bnAbs. And to do that, you needed to know exactly what you were targeting on the virus. This envelope protein, INV. Why is understanding its structure so vital? Right. The INV protein is basically the only thing on the virus surface that these neutralizing antibodies can actually grab onto. It's the key target.
Okay.
But for decades, getting a clear picture of N was a massive headache for scientists. It's notoriously unstable. It kind of wobbles and changes shape constantly.
Like trying to take a picture of something blurry and moving.
Exactly. Imagine trying to design a key, the antibody, when the INV protein keeps shifting its shape. Really, really hard.
So what changed?
Well, huge advances in protein engineering and structural biology techniques like cryo-electron microscopy finally allowed researchers to stabilize N and see its structure in incredible detail, high resolution.
And IAVI NEC was involved in that too.
Oh, yes. Seminal papers came out from IAVI's NAC investigators and their partners globally, Kenya, the Netherlands, the U.S., really mapping this out.
And what did seeing that structure actually allow them to do for vaccine design?
It gave them the blueprint. By seeing exactly how the bnAbs latched onto N, they could identify the specific spots on the virus that were vulnerable. The Achilles heels, if you like. This detailed knowledge was absolutely fundamental. It allowed them to start designing vaccine components, immunogens, specifically engineered to mimic those vulnerable spots and train the immune system to attack them.
That makes total sense. So you finally see the target clearly, you know where the weak spots are, which leads us to this really cutting-edge approach, germline targeting. Can you break that down?
Yeah, absolutely. So with the knowledge of bnAbs and the N structure, the dominant idea now is that an effective vaccine probably has to get the body to make its own bnAbs.
Right.
And germline targeting is IAVI's leading strategy developed with their partners to try and achieve exactly that.
Germline. What does that refer to?
Germline refers to the very naive precursor B cells in our immune system. These are the immature cells that have the potential to eventually mature into antibody-producing cells.
Okay, the starting point for antibodies.
Exactly. The idea of germline targeting is to use these specially engineered immunogens in a very specific sequence, like a prime shot, then maybe several boost shots.
A multi-step process.
Yes, a very careful sequence designed to guide these specific naive B cells down a particular developmental path. Step by step, you're essentially coaching them, training them to mature into cells that can produce the desired bnAbs.
So you're not just showing the immune system the final target, you're teaching it how to build the weapon to hit that target.
That's a fantastic analogy, yes. You're training the antibody factory from the ground up.
And has this approach shown any promise in trials?
It really has. IAVI's first engineered immunogen using this approach, called EOD-GT860-MER, went into a phase 1 trial, and it worked. It successfully activated the right kind of precursor B cells, the germline B cells they were targeting. It was the first time vaccination had ever been shown to do that for bnAb precursors, a huge milestone.
That's incredible news. First step achieved. So what came next? You mentioned Moderna's mRNA tech.
Yes. Building right on that success, they launched a follow-up phase 1 trial, IAVI-G002. This tested a second engineered immunogen called Core G28V260-MER.
Designed to do what?
Designed to take those B cells activated by the first shot and nudge them further along that pathway, guiding them closer towards making actual bnAbs.
Okay, the next step in the training.
Exactly. And what was really innovative here was that both the first immunogen, EOD-GT8, and the second one were delivered using Moderna's mRNA platform.
Like the COVID vaccines.
Precisely. Same technology. The mRNA gives your cells the instructions to make these specific HIV immunogen proteins.
A really clever application of that tech. Was that trial just in one place?
No. And that's crucial. Recognizing that HIV affects the whole globe, particularly Africa, they ran a companion study, IAVI G003.
Okay.
This trial tested that first immunogen, EOD-GT8-16mer, again using the mRNA platform, but specifically in African populations Rwanda, Kenya, South Africa. And importantly, it was led by African scientists through the ADVANCE program.
That's great. Emphasizing global collaboration and relevance.
Absolutely essential. And we're all keenly waiting for the results from both G002 and G003. They're expected sometime in 2025.
So the bigger picture, the long-term goal for IAVI is what? A whole series of these shots.
And that's the idea. Develop a sequence of vaccines, maybe several shots with different engineered immunogens, that together can stimulate the production of multiple types of bnAbs.
Why multiple types?
Because HIV is so diverse. Hitting it with several different kinds of powerful antibodies gives you a much better chance of covering most strains and preventing escape, aiming for a broad, potent response.
Makes sense. A multi-pronged antibody attack. Now, bnAbs sound amazing, but you also mentioned T cells earlier. The scientific consensus seems to be we need those too. Why?
That's a really critical point. While bnAbs are great for, hopefully, stopping the virus from getting into cells in the first place.
The first line of defense.
Exactly. But many scientists believe a truly effective vaccine will probably need T-cell responses as well, particularly cytotoxic T lymphocytes, or CTLs.
And what do they do differently?
Their job is different. They recognize and kill cells that have already been infected. So if some virus slips past the antibodies.
The T cells are the cleanup crew, the hunters that take out the infected cells.
That's a perfect way to think about it. They mop up any infection that gets started, controlling the virus after entry. So bnAbs are the shield, T cells are the internal security force.
Okay, so you likely need both for robust protection. Is IAVI working on T cell vaccines too?
Yes, absolutely. They're pursuing T-cell vaccine approaches in parallel with the bnAb work. Recognizing that combination is probably the way forward.
And what do those T cell approaches look like?
Well, one key strategy is developing immunogens based on parts of HIV that are conserved, meaning parts of the virus proteins that don't change much across all those different strains. They stay relatively consistent. So T cells, trained to recognize these conserved regions or epitopes, should work against a wider variety of HIV variants.
Similar logic to the bnAbs, finding common ground, but for T cells.
Exactly. And IAVI supported a phase 1 trial of one such conserved immunogen, which actually finished up in July 2023. We should see results from that fairly soon.
Interesting. Any other T cell strategies?
Yes, researchers at IAVI's own vaccine design and development lab are exploring other platforms too. One involves using a different harmless virus, a recombinant vesicular stomatitis virus, or VSV vector.
As a delivery truck.
Sort of, yeah. It's modified to carry a piece of HIV genetic material, in this case, part of the N gene, into cells specifically to stimulate a strong T-cell response. That's still in preclinical studies, testing safety and potential effectiveness.
Okay, so really exploring multiple avenues there too. It definitely sounds like the strategy is attack from all angles.
It really has to be, given how complex HIV is. A multifaceted approach targeting both antibody and T-cell immunity seems the most promising path.
Well, this has been incredibly insightful, a really fascinating deep dive into this huge challenge of finding an HIV vaccine. It's clear that despite, decades of difficulty, real tangible progress is being made now, especially with this focus on bnAbs and T cells.
Absolutely. That germline targeting approach, it's genuinely innovative science. And the early results, especially combining engineered immunogens with mRNA tech, it really does offer, well, cautious optimism, a significant step forward.
Definitely feels like a different phase of the research now. And it's just so important to remember this isn't happening in isolation, is it?
Not at all.
It's this massive global effort. Scientists, trial participants, Africa, Europe, America, Australia, all working together.
It's a fantastic example of what international scientific collaboration can achieve when focused on these major global health problems that ultimately affect everyone.
Yeah. So as we wrap up, it's clear the road to an HIV vaccine is, well, it's a marathon, definitely not a sprint.
Absolutely.
It really highlights how complex scientific discovery can be and the sheer persistence needed to tackle something like HIV. Kind of makes you reflect on the scale of these efforts and just how vital continued research and that unwavering commitment really are.
Well said. The dedication is incredible, and it has to continue.